Hello, I am currently at the Harald von Boehmer Midwinter Conference in Seefeld in Austria, and I thought I would record my talk on tissue regulatory T cells or tissue Tregs for people who weren't lucky enough to be here this weekend. Okay, so today I'm going to be talking about regulatory T cells in the tissues with a particular emphasis on can we use these regulatory T cells in the lung in order to reduce pathology. Regulatory T cells have now been identified in most tissues as a population that is resident inside the tissue. Now, as well as having the classical properties of most regulatory T cells, being the ability to shut down immune responses, these regulatory T cells inside the tissues also have functions in promoting tissue repair, aiding tissue homeostasis, and in promoting tissue regulation at a more global level. Now, the latest data for regulatory T cells in the tissues indicates that these cells are not really long-term permanent residents, and instead they have a more transient residency pattern. So the regulatory T cells that are present in circulation, they're able to enter into tissues. They turn on a tissue program, a transcriptional rewiring of the regulatory T cells that differentiates them into this tissue phenotype. But really, it's a pretty generic program. So the program that you see in the lung is very similar to the program that you see in the liver because it's really only coming on for a few weeks to a few months before that cell is going back into circulation and is going to possibly be migrating through multiple different tissues over its course over the lifespan. So it looks like these regulatory T cells that are present in the tissues really have a pan-tissue lifestyle, with a potential exception for a subset of the regulatory T cells present in the gut that have got a distinct phenotype and different tissue homing patterns. Now, we're quite interested in knowing what the molecular requirements are for a regulatory T cell to get into the tissues, and once they're in the tissues, to adapt to that tissue lifestyle. And there are multiple studies coming out by different groups now that are taking a candidate-based approach and identifying whether or not that candidate transcription factor or that candidate chemokine receptor is able to change the ability of the regulatory T cell to get into the tissue. What we're quite interested in doing, though, is taking a more global look, a systematic analysis of entry factors and transcriptional rewiring factors to see which of these are impacting tissues at a global level and which of these are impacting specific tissues with the ability of regulatory T cells to get in and to acclimatize to that tissue. And actually, it's probably a tractable problem. There are a relatively limited number of genes that are involved in migration from the side of the migrating cell. If we take all of the integrins, adhesions, cadherins, selectins, chemokine receptors, all up, it's about 150 genes. And if we look at the transcription factors that are involved, there's only about 50-odd transcription factors that are upregulated in tissue Tregs across the body. So there's a relatively limited number of candidates that could be involved in this process. Now, in principle, we could test this using a Treg transfer system. We make a knockout mouse, we purify knockout cells, and we purify wild-type cells, we mix them together, and then we transfer them into a new mouse, and we could look at the ability of the knockout cells to get into tissues versus the ability of the wild-type cells that were co-transferred to get into the tissues. You simply have to make 200 different knockouts, do 200 transfers, multiple mice, screen all the genes for tissue entry. The only problem really is about logistics. If you look at the number of tissues involved, the number of groups involved, you'd need about 4,000 mice and 68,000 flow cytometry files, and it's just not practical to run. Essentially, in order to run this type of experiment, you need to be able to do massively parallel barcoding experiments in vivo. Now, fortunately, we're able to build on the work of Brian Brown's lab, where he developed a system for massive parallel experiments. This was first done in vitro in this really seminal Cell paper. And essentially, he used a CRISPR-Cas9 approach to knock out multiple different genes. So you can take Cas9 expressing cells and you can use a lentivirus to introduce the guide RNA in order to knock out individual genes at a time. Now you can use a library of lentiviruses, each expressing their own guide RNA, so you can create a mixture of knockout cells where each cell has got a potential different knockout. Now the only problem with this library approach is that you need to be able to know which cell carries which lentivirus so that you know which cell it has a knockout for which gene. Now the approach that the Brown lab did was to use CyTOF, where they could measure which lentivirus was being introduced to the cell based on that lentivirus carrying a marker that could be detected by an antibody. And you can imagine in a system where you can introduce a red marker, a blue marker, and a yellow marker, you can mix all of those cells together and you can easily say which cell is a knockout for gene 1, gene 2, or gene 3 based on does it have the red marker, the yellow marker, or the blue marker. Now the problem is that when you start to go very high dimensional, you run out of single markers that you can use. And the real insight of the Brown lab was that you're not constrained by single markers. You can create combinatorial effects by, for example, using a triplet marker. And if you use 14 different markers and combinations of triplets, you end up having nearly 400 different combinations of those markers. That means that you can have a mixture of lentiviruses containing 400 different guide RNAs to knock out 400 different genes, and each of those lentiviruses will have a different triplet combination of 14 basic antigens. So just by measuring 14 antigens with antibodies, you can work out for every single cell, is it a knockout for gene 1 up to gene 200. It's a really great approach and it works very nicely in vitro. There's just a problem in vivo that when you're transferring these cells, the initial codes were encoded on the surface and you end up having quite a lot of transplant rejection issues and you can't really pick up the codes by flow cytometry. So we modified this system, really some great work from postdoc in the lab, Adrian, and we're using exactly the same antigenic tags as the Brown lab. It's really deriving those same antigens. We're cloning them into a retrovirus, and now we're fusing them to an intracellular protein, histone 2b. Now the advantage of fusing to histone is that now it becomes an intracellular tag. It prevents rejection when you're transferring the cells in vivo. The tags build up to much better levels for detection by flow cytometry, and you can also have intracellular staining without losing the tags because the tags are really fixed onto the genome through fusion to histone. And we've now validated this approach in vivo with flow cytometry. So we've got a system now where we can put our guide RNAs into these retroviruses. We can have up to 400 different retroviruses if we want. Each of them has their own guide RNA. Each of them will knock out their own gene, and we can track which retrovirus any individual cell has by looking at which triplet combination they have of these 14 different antibodies. And so really you can do flow cytometry. You just need to have 14 markers to pick up your flow codes, and then you can use the other channels will be available to look at any phenotype markers you're interested in. So this is how the basic experiment is set up. We can take Cas9 positive cells from a Cas9 transgenic mouse. We can purify those T cells, and then we can put on a retrovirus from the flow codes that has a library covering all of the integrins, adhesions, cadherins, and so forth. And so you've now got a mixture of these 150 knockouts, and we can track them all by flow cytometry, and we can move that entire mixture into a RAG knockout mouse and then look for it to reconstitute the population of the tissues later on. And we can then simply do a spectral flow cytometry analysis of the different tissues to look at which knockouts are finding it difficult to get into particular tissues and to become tissue-resident regulatory T cells. So this is some analysis done by a talented bioinformatician in the lab, Vaslav, and it's the type of experiment that we're doing now with the flow codes. So we've got 10 mice, 18 tissues, so it's a big experiment of 180 flow cytometry files, but this experiment includes data from all 150 potential migration genes, all the integrins, all the chemokine receptors and so forth, and we're covering 18 different tissues. And so for each of these 150 genes, we've got the phenotype of the knockout cells, and we've got information on whether or not those knockout cells are able to get into each of multiple different tissues. We can also look at CD4 cells, CD8 cells, regulatory T cells in the same experiment. So this is some data from regulatory T cells. We're looking at multiple different genes, we're looking at multiple different tissues, and these are all of the effects that are statistically significant in this cohort. You can see the bottom couple of rows are the gut. They've got a seemingly different set of migration genes that are involved. This blue streak along the top is the bone marrow, much more enriched for chemokine receptors. And then the genes in the tissues in the middle are the classical non-gut, non-lymphoid tissues. And we can see a couple of hits coming up. And then this is the data if we don't have a significance filter. So there are sometimes genes that are moving in the same direction, just not hitting the significance filter. So an example of this would be to look at individual tissues. So what controls the entry into the lymph nodes versus the spleen, or entry into the gut versus the spleen. And we can do this type of analysis. And if you look at lymph nodes versus the spleen, you have a very simple entry system where the main entry requirements to get into the lymph node are the integrins that are part of LFA1. And this makes a lot of sense to us. There are monoclonal antibodies that are available that block entry of T cells into lymph nodes, showing that this target is validated and cross-species as well. Now, if we go into the gut, we see a different set of genes coming up on this volcano plot. Here we've got alpha-4 beta-7 being important for T cells to get into the gut. And again, this makes complete sense because we've got a monoclonal antibody that is used in the clinic and it blocks T cell entry into the gut. So it's conserved and it's hitting biologically relevant pathways. Now, how about transcription factors? Now, here again, we've got this single experiment where we've got 55 different transcription factors knocked out. We're looking at multiple different tissues, and we can see which of these transcription factors, when you knock them out, give you a significant defect in getting into a particular tissue. Now, the gene that comes up the most commonly is that of BLIMP1. It is required for entering the brain, the kidney, the pancreas, the skin, the female reproductive tract, and the testes. And now if we show all of the hits and not just the significant hits, you can also see that it impacts lung Tregs and adipose Tregs. So BLIMP1 really seems to be a pan-tissue Treg requirement. All of the Tregs in all the tissues essentially need BLIMP1. We can also look at particular tissues. So here we're looking at skin versus spleen. And if we look at what Tregs are required to become a skin Treg, we are getting hits like BLIMP1 and BATF, which seem to be pretty global and important in multiple different tissues. And then we have hits like GATA3, which really is coming up very strongly for skin Tregs and doesn't come up very strongly for other tissues. So that might be disproportionately important for skin Tregs as opposed to other tissues. Now, in the second part of the talk, I want to go through whether or not we can harness regulatory T cells in the tissues to prevent pathology. For example, can we use lung Tregs to prevent lung pathology? And this was a collaboration with our colleagues at KU Leuven in Belgium and led by a talented PhD student, Tomby. Before I get into the science of the project, though, I want to spend just a couple of slides talking about some of the technical limitations. And this is because using spectral flow cytometry in the lung is quite challenging, especially if you're using a ultra high dimensional panel. Now, it's generally true that ultra high dimensional panels frequently have fluorescent assignment errors. We have issues of skewing, issues of spread, we have suboptimal spillover calculation, and a lot of this is derived from fluorophore heterogeneity and autofluorescence heterogeneity. Now, this is true in any flow cytometry panel once you start going up and hitting the limits of the number of markers you can use, but it's especially problematic in tissues such as the lung, where you have multiple different cell types with completely different autofluorescent signals. And when you're deconvoluting the data, that autofluorescence goes into different channels, making it very challenging to do good flow cytometry on a tissue such as the lung. It is really important to note, though, that this is not a hard constraint based on the hardware. This is not an issue that is unresolvable in spectral flow cytometry. And in fact, the issues that are generated seem to be generated in the majority of cases by data processing approaches rather than based in the hardware. So you can take the raw data and end up with much better processed data if you're simply improving the statistical methods that you're applying to unmixing flow cytometry. So to do this, we've developed a program, AutoSpectral. It's not published yet, but there's a link here and a QR code. And there are multiple different aspects to AutoSpectral. Now, one thing that AutoSpectral does is it takes the place of a skilled flow cytometry analyzer. So it will do an automatic selection of the ideal unmixing populations. This involves scatter matching and removal of intrusive events. It's the type of work that a really skilled flow cytometry analyst can spend hours and hours doing, days even, and it improves the outcome. But we just replacing all of that manually with a computational approach. Now, the second thing that we do is we apply robust linear models to optimize the unmixing. This is something that we developed a few years back in AutoSpill, and is now in essentially every commercial software for flow cytometry analysis. And it reduces the error of unmixing or compensation by optimizing that assignment of spectrum to different channels. That approach that we published in AutoSpill also allowed you to unmix one autofluorescent channel and improve the results that way. But a limitation is that it was really based on one autofluorescent channel. Now, in our new software, AutoSpectral, we have got the ability to do autofluorescent matching and spectral spectrum matching at an individual cell level, which means we can deal with multiple different autofluorescent signatures. Essentially, what we can do is we can use the unstained sample and identify all of the different types of autofluorescent signature that are present inside that sample. We can then unmix the data from a stained sample, and for each individual cell, we can ask, does this cell give you less residual if you unmix it with autofluorescent profile 1, autofluorescent profile 2, or autofluorescent profile 3? And it does that for every individual cell individually, which means that at the end, you have got something that's been unmixed using the correct autofluorescent spectrum for that particular cell. Now, this gives you about a 9,000-fold reduction in the misassigned spectrum, and it is really night and day when you analyze tissues like the lung that have got very different signatures that normal software approaches just can't handle. And this is an example of the type of data you get on the lung if you're unmixing with current state-of-the-art techniques versus if you're unmixing with AutoSpectral, and a lot of those artificial signatures have just disappeared. Okay, I'm going to go back onto the science now and talk about the role of tissue Tregs in the lung. Now, when we're studying regulatory T cell functions in the tissues, one of the ways that we can study them is to expand the population up to higher levels. And a good way to do this is to supplement the key food for regulatory T cells, which is IL-2, and boost that up in amount in a particular tissue. This essentially boosts up the niche size for the regulatory T cells so that when they go into that tissue, they can survive longer, they can proliferate a little bit more, and you have got a boost of the number of regulatory T cells inside that tissue. Now, IL-2 is very localized, it doesn't travel very far, which means that you're not getting any effect on the global immune system. The number of regulatory T cells in the draining lymph nodes is completely intact. The number of regulatory T cells in other tissues is completely intact. And so it allows you to really say, what is it that the regulatory T cells are doing in the tissues that is distinct from what they're doing in the lymph nodes and the rest of the body. Now, we can deliver this IL-2 into different tissues using an adeno-associated viral vector-based delivery system coupled with tissue-specific promoters. And a proof of principle of this was our use of the PHPB GFAP promoter, where we could deliver IL-2 to the brain and boost regulatory T cell numbers in the brain only. Now, in this current study, what we've done is we've developed a new AAV system. It's an AAV 6.2 system using the CC10 promoter. And under this system, we get production by club cells in the epithelium of the lung. We get production of IL-2 inside the lung. We've got no extra IL-2 in the draining lymph nodes. Our regulatory T cells expand in the lung. No expansion on the draining lymph nodes, no expansion in any other organ that we've looked at. We've used this system to boost IL-2, but we've also shown that you can use it to give you lung-specific expression of other cytokines such as IL-10 or IL-1RA. So we've got this system now, and we can boost regulatory T cells just in the lung tissue while leaving the rest of the body normal. Now we want to look at what happens in a challenge situation. And the most relevant challenge situation that we could come up with was this context of influenza-associated pulmonary aspergillosis. This is really the primary killer of the flu. When people are hospitalized with severe flu, the mortality is often not just driven by a flu monoinfection, but is really driven by this influenza aspergillosis co-infection, which is really quite striking because Aspergillus as a fungus is a pretty harmless infection by itself, and it's only under cases such as flu infection or other immunodeficiencies where it becomes such a killer. But when you do have the co-infection, the mortality can be over 50%. So our system here is a mouse model where we can give mice the flu, and then on day 4 of the flu infection, we give them the Aspergillus fungus, and we have our primary takedown on day 7. Now here we've got some mice that have just got the co-infection. This is IAPA, the influenza-associated pulmonary aspergillosis, or they have IAPA plus IL-2, where they've got extra regulatory T cells in the lung. Now this is data showing weight loss, and of course uninfected mice are quite healthy, whereas influenza-infected mice are losing weight, about 15% of body weight. If we have the co-infection, we're losing more like 25% of body weight, and these mice will reach a level of sickness where we have to put them down. But if we've given them the co-infection and we've boosted the regulatory T cell number, then we're removing about 50% of the extra body loss. So we're protecting the mice from a lot of the extra body weight loss. We can also measure the health loss looking at the cumulative health impact score. So again, mice with influenza get sick, but mice with the co-infection, they have a much higher health impact score. And if we are giving the mice the co-infection and we're boosting the regulatory T cell number, we protect about 90% of our added health loss. So we turn a really lethal infection into a regular flu infection. We can also measure the amount of lung damage using non-aerated lung volume. And again, what happens is the influenza has got lung damage, the co-infection has got much worse lung damage, and the co-infection plus extra regulatory T cells in the lung reduces that added damage by about 80%. For neutrophilia, the protection is a bit weaker, so we're reducing about 40% of the extra neutrophilia that is observed in the bronchoalveolar lavage. Now, all of this is happening while the pathogen load is fairly unchanged. We can measure either influenza in the middle graph, or on the right-hand graph, we can measure aspergillosis, and we see really no impact on influenza load if we are adding extra IL-2 into the system, extra regulatory T cells in the system, and we also see only a mild reduction of aspergillosis load in the mice that have extra regulatory T cells. Certainly, we are not seeing a situation where the infection is unleashed. And I think this is probably because the draining lymph nodes are completely normal. The amount of anti-influenza antibody that is made is completely normal. The number of primed CD4 and CD8 cells against influenza is completely normal. So we're not shutting off that adaptive immunity happening in the draining lymph node, we're just shutting off the local inflammatory processes occurring in the tissue. And this is a summary graph which really shows that we're able to improve on multiple different health measures. We're getting a really fatal inflammation down to a normal influenza infection rate without actually changing the infectious load much at all. So with that, I want to finish up. I really want to thank the amazing team members that were involved in this, in particular Tomby for leading the lung Treg project, Oliver for developing autospectral, Vaslav for leading the flow code analysis, James Dooley, everyone else in the lab. I also want to thank the amazing collaborators we've had, in particular the Belgian collaborators with the IAPA model. I've got a link here for career advice articles for any early career researchers that are watching, and also a link for autospectral if you're interested in adopting our new flow cytometry software. And then finally, a thanks to the ERC and the Wellcome Trust for funding us for many years. Thank you.